Dear all, a very good day. Today we are going to discuss or we are going to continue forward the topic of last class that was about the electrodes. Now we are going to have a discussion about biochemical transducers. Okay. We have already discussed what a transducer is. So the method for measuring the concentration of ions or gas uses normal method is to use one electrode, that is an active electrode, which is very sensitive to that gas which we are measuring, and with the help of this active electrode we use a reference electrode. The reference electrode is a standard electrode which is insensitive to that particular substance. Okay. Previously we had discussed that reference electrode is made up of hydrogens normally, which was a standard utilization. So hydrogen gas or ion interface has been designated as a reference interface and was arbitrarily assigned as an electrode potential of 0 volt. So hydrogen electrode are used as reference electrode in biochemical measurement. However, there are some disadvantages related to this type of electrode. These electrodes make the use of principle that an inert metal such as platinum readily absorbs hydrogen gas. If it is properly treated, piece of platinum is partially immersed in a solution containing hydrogen ions, okay, and is also exposed to H gas, which is passed through the electrode and electrode potential is formed. Okay, the electrode leads to the lead is attached to the platinum. Okay, but a hydrogen reference electrode is not so simple and stable. This is the basic disadvantage of this electrode. Okay, so the other two types of electrodes which we use are silver chloride electrode and Calomel electrode. Now we are going to look at the basic configuration of a reference electrode. In this figure we can see there is this is a structure of reference electrode in which there is an internal AgCl wire or a Calomel wire. This is another type of material. Then there is a filling solution, normally KCl. This solution connects this electrode with the liquid junction of human body. Okay, so the ionic AgCl side of the interface is connected to the solution by an electrolytic bridge, which this is a electrolytic bridge KCl which is functioning here, and is usually dilute solution in the form of liquid junction with sample solution. Now the electrode can be successfully employed as a reference electrode if the KCl solution is also saturated with precipitated silver chloride. Okay, the electrode potential for AgCl reference electrode depends upon the concentration of KCl, how much concentration we are using. If you are using 0.01 mole of KCl, then potential is 0.343, and if we are using 1 mole of KCl, then potential developed is 0.236 volt. The next electrode comes Calomel electrode. It is made up of mercurous chloride, that is AgCl material. The interface between the mercury and mercurous chloride generates an electrode potential. Okay, so whenever mercury and mercurous chloride are connected, they will generate a potential. By placing Calomel side of the interface in KCl solution, an electrolytic bridge is formed with the sample solution from which the measurement can be made. So if we will use a Calomel, that is the silver side of the chloride side of the interface in the KCl solution, so it can form a bridge between the samples, okay, in which solution can be used for measurement. Okay, the measurement values are 0.01 mole of KCl produces 0.388 volts and 3.5 mole of KCl produces 0.247 moles for Calomel electrode. Now, then there is another very important electrode which is the pH electrode, that is the electrode which can test acidic or basic nature of different solutions. The pH is directly related to the hydrogen ion concentration of a fluid. We know that if a hydrogen concentration is very high, then it will be an acidic solution, and if OH concentration is high, then it will be a basic solution. So it is the logarithm of the potential, it is a logarithm of the reciprocal of H plus ions. That is how the pH is defined, and the formula is given here, where pH is negative log 10 of H positive ion, or you can say log 10 of 1 by H positive ion. Now the pH is measured as the acid base balance of a fluid, and a not normal, or you can say a neutral pH solution is of the value 7. Now our blood is slightly basic, and it ranges in 7.38 to 7.42 range in the pH scale. So here is a simple diagram which shows how a pH meter works, or you can say the diagram of a proper pH meter, in which this is the lead which is connecting to the meter. There is AgCl wire contacts, and then there is a pH gas, and there is a buffer solution. Okay, with the help of this we can measure half cell or half plus half cell, indicating the pH of the material or whatever we are measuring. Okay, then inside the glass bulb is a highly acidic buffer solution. In the previous diagram we have seen that there was a glass bulb, so that bulb contains a highly acidic buffer solution. Measurement of the potential across the glass interface is achieved by placing AgCl solution inside the glass bulb and Calomel or AgCl reference electrode in the solution. So we can use AgCl solution, and we can put in this Calomel electrode or an AgCl reference electrode in the solution, in which pH is to be measured. The thin glass membrane allows the passage of only hydrogen ions. So the membrane which we are seeing in this figure, the glass membrane, okay, this membrane is semi-permeable. This membrane is semi-permeable only to our H positive ions, and therefore pH can be measured with its help. Thereafter, there is blood gas electrode. These electrodes are used for measurement of concentration of oxygen or carbon dioxide in our human blood. Okay, so if we want to measure their concentration in the human blood, we have to measure their partial pressure. Okay, so partial pressure of a gas is directly proportional to the quantity of that gas in the blood. So the effectiveness of a respiratory or a cardiovascular system can be measured by measuring the partial pressure of either oxygen or carbon dioxide. This will tell us the capability of our blood to carry these particular gases to our different cells. So oxygen, the basic principle for the measurement of oxygen tension is that a platinum electrode is used, or some other noble wire metal is used. Okay, and it is embedded in a glass for insulation purposes, with only tip exposed to place an electrolyte into which oxygen is allowed to diffuse. Okay, so oxygen from the blood can be allowed to diffuse. So this is some sort of a setup which has been shown, in which this is AgCl reference. This is the electrolyte solution in which our O2 can diffuse. Now this is the membrane through which O2 will diffuse. So this is a semi-permeable membrane for oxygen gas. Okay, so if a voltage of 0. And this is an insulating electrode here with a platinum tip. So this is a platinum electrode over which we have provided an insulation. So if a voltage of 0.7 volt is applied between the platinum wire and the reference electrode, with platinum wire negative wire with negative potential, so reduction of O2 takes place at the platinum cathode. So reduction process takes place at the platinum electrode. As a result, an oxidation reduction current proportional to the partial pressure of the diffused oxygen can be measured, and in this way measurement of oxygen is achieved. The other important gas whose concentration we need to measure is the partial pressure of CO2, because CO2 is very harmful and it is almost as harmful as carbon monoxide. So there is a linear relationship between the logarithmic for partial pressure of CO2. This is PCO2 is partial pressure of CO2 and the pH of a solution, because CO2 when mixed in liquid easily generates our carbonates. Okay, so measurement of partial pressure of CO2 is accomplished by surrounding the pH electrode membrane with selectively permeable CO2 membrane. So like in this figure we have a selectively permeable oxygen membrane. Instead of this we will be using selectively permeable CO2 membrane. Okay, then there is a Severn-Gauss electrode in which membrane permeable to the CO2 is made up of Teflon. Here is a structure which we can see of the basics arrangement for CO2 measurement, in which semi-permeable membrane of Teflon material is used, and it does not permit any other ion to pass through it that might affect the pH. So here is a thermostated matter which can be connected. Then there is a glass tube here. This is the electrolyte. There is a platinum wire for measurement of partial pressure of CO2. This is the reference electrode. This is the stainless tube, and different materials can pass through here. And then there will be Teflon coating which can only pass CO2. So the space between a Teflon and the glass contains a matrix consisting of thin 0,0 shear nylon which is used. This matrix serves as support for an aqueous bicarbonate layer into which CO2 gas molecules can diffuse. So only CO2 gas molecules can diffuse. So measurement can be of partial pressure of oxygen or CO2 can be combined into a single electrode as well, which includes the common or reference half cells. Okay, thank you.